There's a saying in medicine, the right treatment for the right person at the right time. But what if that applied not just to diseases, but to aging itself? What if we could not only slow aging, but do it precisely, tailored to each individual's biology, lifestyle, and risks? Hey everyone, welcome back to Decoding Longevity, where we break down the science of aging and longevity. I'm your host, Raghav Sehgal, joined by my co host, Macsue Jacques.
Hi, everyone. In our last episode, we have explored the new pioneering work in epigenetic clocks and the new ensemble age model, and how these tools might one day help us to measure and track biological aging in real time. But today we're taking the next big step. How do we actually use this information to actually manage aging in the clinic? For this, we are diving into a very new landmark paper published in Cell called From Geroscience to Precision Geromedicine and Understanding and Managing Aging. And to help us break down this paper, we have one of the authors, and I'm a big fan, to Professor Andrea B. Maier, a world-leading geriatrician, physician, scientist, entrepreneur, co-director of the Center for Health and Longevity at the National University of Singapore. So, Andrea, welcome to the show.
Thank you so much, both of you.
So, Andrea, before we get into the details, can you help us set the stage? What exactly do you mean by precision geromedicine, and how does it build on geromedicine itself?
So, precision geromedicine is a new term we coined a couple of months ago, and for the first time we used in our Cell paper. It actually is the academic word for healthy longevity medicine, and healthy longevity medicine was coined by the Healthy Longevity Medicine Society in 2023, where we said healthy longevity medicine is optimizing health and health span by antagonizing aging processes across the lifespan. So that's a new discipline we want to introduce into conventional healthcare systems to optimize the health of aging individuals by really targeting the aging processes. Very often I heard, what is healthy longevity medicine? It's not Greek, it's not Latin, so it has nothing to do with medicine. What is healthy longevity? Hence, we said, okay, we have the term gero. Gero means aging or age, depends on what school you have, being traveling towards to. Geromedicine is nothing else than aging medicine, and precision is being precise and being accurate in an NS1 approach that we can optimize the health for individuals.
Yeah, that is very incredible. I love the N of one approach and going into the precision medicine. That was like my whole sort of like back on my PhD days where trying to measure that inter-individual variability. So I'm so happy that it's finally coming into the longevity field as well. So in a way, I think we're moving from aging science in the lab to aging medicine in the clinic, right? So can you give us a practical example? I know that you have so much background in the clinic, being, a doctor in practice. So what is actually looking like to the patient that comes to the clinic and is receiving now this personalized advice?
Yeah, before giving advice, very importantly, we have to diagnose something, because that's what we normally do in medicine. Patients are not coming to me and say, I want chemotherapy. That's not how it happens. No, first we are diagnosing cancer, then we're giving a therapy. The same for diabetes medication. We don't prescribe it without having a diagnostics in hand. And I think there is where our field, precision geromedicine, really has to catch up. Our entire field has to catch up, because we as physicians very often, and the consumers especially, are hunting for that therapeutic. And I think first we need to know what is the biological age of individuals, what is the pace of aging of different organ systems, what is the real pain point for that individual, which we should actually tackle. How many of the other organs are already compensating for the aging process of other organs? So first we have to understand what the aging process looks like in that individual, and before going and hunting for any intervention. So in my clinical practice, but also this is the recommendation of the Healthy Longevity Medicine Society, we have 3 different pillars of how to measure the aging process of individuals. And we do that by applying biomarkers of aging, which are defined by the Biomarkers of Aging Consortium. So we have biological biomarkers, we have clinical ones, and we have digital ones. And all 3 of them in combination are being applied in clinical practice, but also from research. And of course, research is feeding the entire chain of bringing it into clinical practice. If we're talking about biomarkers of aging being biological, we can talk about genes. What kind of SNPs does somebody have? We can talk about epigenetics. We can talk about the microbiome. We can talk about other lab values, which are also biological. Think about a testosterone level. If we are talking about the clinical phenotypic characterization, we can measure individuals from hair density to toenail lengths and density. So, but in between, we have the heart, the liver, the lungs, the brain, and everything needs to be, in my view, measured to get the real understanding where that person at the moment stands in terms of the aging process. So think about not only imaging of the brain, but especially measuring the function, measuring the cognitive function. The same for the heart. We can image things, but we can also functionally measure. We can stress individuals on a treadmill, on a bike, doing a VO2 max. And there was looking at the interaction between the lung, but also the cardiovascular system next to motivation, which is also important. Then we have the digital markers. The digital markers, hopefully everybody wears already digital devices and gets health clues out of it. But not only thinking about something we wear about our fingers and our wrist, we now also have continuous glucose monitoring systems. We have other devices which are now ready for implementation into clinical practice to really measure real time, continuously, the physiology of individuals in individuals' homes. And then next to that, combining that with the motivation of individuals, with the psychology behind it, and that glued together and intermingled together is really then leading towards a diagnosis, and if people want, towards very precise interventions.
Yeah, I think you've really beautifully put this, Andrea, and especially sort of laying out all the different biomarkers that are out there potentially that could be used for precision geromedicine. So I guess I have a question here for you, which almost goes back to the age-old problem in the field, which you sort of point out in the paper as well, which I would say is in some ways almost like an update on the hallmarks of aging. There is a lack of consensus within scientists on what aging itself is, right? And I would love to hear your thoughts on this. Are we really ready to move beyond the complexity of saying what is aging and then moving towards precision in aging?
So, first of all, we need a definition for aging. I think if we are looking at our field and we are calling ourselves aging researchers and we haven't defined aging, I think there is a lack. I'm arguing for that since a long, long time. I would love to just put people into a room and then just until we have the white smoke, nobody is going to leave. And it's not about if it's the right definition, but it's about which definition we are using. And that's very different. If it's right or being used, very different things. I would love to see, okay, this definition is now being used until the next consensus meeting, until there's white smoke. And then we can change it. And in the meantime, in parallel, we can actually then emerge in our knowledge until we have a better solution. So we need that. I'm a physician. I'm, of course, a researcher. I love to understand the biology. But for me, if I explain it to my patients, it's the accumulation of damage, and it occurs all the time. It depends on how good you are actually to remove the damage. You will accumulate it.
Yeah.
But it's reversible, because if your house is not clean and you bring a cleaner in or you do it yourself, you can have and invite friends again. So that's the same analogy. And of course, some of the damages which occur might not be reversible. So here is, I think, the starting sign. Okay, you might want to act now that you are not accumulating the irreversible damages too much. So how do we define? That's my definition. Very important. If we are talking about definitions, we also have to be able to operationalize it. Because very often we have definitions and you say, oh, that sounds beautiful, but so what? What do we do with it?
Yeah.
And I think there, the biomarkers of aging consortium really comes in, where we are now meeting and say, okay, we can define what epigenetic clocks are and we can describe it. But how do we operationalize it, either in research or in clinical practice? And is there the prime time already for clinical practice implementation? And that's actually what we are doing right now. There will be a consensus statement from the Health and Longevity Medicine Society and from together with the Biomarkers of Aging Consortium, if and how to use epigenetics in clinical practice, and if there is the prime time already. And I think that's what we need to do. We have to get the information, come together, define it, operationalize it, and release it until the next round and the next version.
Absolutely. I think that's, you touched right on the next question that I have. That's where the other omics start to come from and start to being integrated into this clinic. And obviously epigenetics being one of the most proven time and time again to track really well, not only the chronological age, but also the biology of age. I think it's overdue to be implemented in the clinic, in my opinion. But then we also have, the other omics. We have the proteomics, the transcriptomics. Do you see any of those playing a role as well in building personalized profile, or is epigenetics, in your opinion?
So let's talk a second about epigenetics, because what we very often say, okay, it has been published. There is the association with if you want to predict mortality, a hazard of 1.3, for example, 30% higher odds to die. Clinically, what does it say? So very often we jump, okay, there is this statistically significance, but it does not mean that it is clinically relevant for individuals. So we had huge discussions if epigenetics should actually is at the prime time for clinical implementation. And in the yes, we said a very silent yes, but lots of buts.
Yes.
So despite of all the papers, which very often is coming out of cohort studies, which is very important, having a biobank and then being measured in batches, it doesn't mean that it's prime time for clinical practice where I get a sample now, a sample in 3 months' time, a sample in 3 years' time, where we have to control and cannot control as rigorously as cohort studies, for example, for batch effects. And we already know, and we see it in our own data, batch effects still are huge in epigenetics. So if you're using the EPIC V2 test, between the chips, there's a huge effect.
Yeah.
So the question is it ready for prime time? In principle, yes, but we have huge issues in terms of the methodology being used because of high coefficient of variations in terms of one versus the other measurement across timeframes.
Yeah.
And that's the problem at this moment in time. I would say the intellectual properties we all have in our mind, and we know what to do with the data, but measuring it is most important. Yeah. And also, when we are saying it's ready for prime time, very often we are making use of blood samples in cohort studies where the DNA is extracted from and being measured. However, in clinical practice, if you have a practitioner, that will be difficult to actually send the blood tube, send it to a lab where they are doing the DNA extraction and then doing the epigenetics with a lag time very often, if you're just sending one sample, of weeks or months until you get the result back. Very often in clinical practice, saliva is being used, and we already know that the variation between DNA methylation ages of saliva and blood is huge. And the variation between samples, if you're sampling saliva, is even more huge. It's huge. So there are lots of things and steps to solve, to implement things. So it's not as easy as we think. But I really think, yes, it should be, and we should solve these little but super important steps until we make use of it in clinical practice.
Yeah, Andrea, I think you hit a very, very critical point here, that sort of translation of these clocks that we have, these DNA methylation epigenetic clocks from cohort studies to individual level is gonna be some effort, right? Whether it's the reliability of them on the recurring, on a longitudinal scale, measuring them again and again, will that give the same result? Or will there be variations because of confounders like time of day, so on and so forth? So I really love that you're sort of making this a point in some of your conversations. And I truly believe that this is a very big issue for getting some of these things to clinic.
Mm.
But going back to a bit of the point that you were saying about sort of measuring different readouts, right? Not just sort of clinical biomarkers and omics, but also sort of we have now a lot of biomarkers that can go like in the form of variables. It's almost like you're forming what one would call an aging fingerprint for each person, right? And so, given that you're a physician and a researcher, how close are we actually to sort of forming this sort of comprehensive aging fingerprint for an individual, which goes beyond just looking at a single omics, but multi-omic, as well as looking at sort of different readouts like wearables and omics and the likes of those. And are we even close to sort of implementing them in clinic, right? Are there examples of sort of this sort of multi-omic aging fingerprint approach being used in clinic? Of course, some of the work that you do in clinic does do some of it, but I wanted to know some specific examples where you have seen this.
Yeah, I think we can absolutely improve. I think if you're describing our field and how we build the fingerprint, you call it, I would call it, it's a digital twin, or it's characterizing on different levels the physiology of that individual.
Mm-hmm.
That's what we want to tackle. I think we are absolutely there, but I think we are not adult yet in the adulthood, but we are rather in the toddler stage, where we are trying to get more and more of these markers included, and we try actually to walk. I think this toddler stage could be a very good analogy for the entire field and not just for the diagnostics and how to characterize individuals. But yes, in clinical practice, at least in my clinical practice at Longevity, we are using genetics, we are using epigenetics, we are using the microbiome, we are using the digital phenotypic characterization next to every clinical parameter we can measure in an accurate and validated way. So that's important. I think it's not a problem. I think the beauty at the moment is how do you relate all these different levels of the aging process together? And that's what we are doing, for example, at NU, which is a company I'm the CMO of, where we are building digital twins. We are combining these data based on the best evidence we have, because every step we should do should be evidence-based in my view. And then only we really have that really nice product of an evidence-based digital twin of a human being, which we can then manipulate and also use to actually see if certain therapeutics might help. But in clinical practice, I think we have to make sure that every step we are taking, every marker we are taking, that it is clinically relevant for that physician to be clinically relevant in the end also for the patient or the client that physician is seeing. And some longevity clinics are a little bit further than others. We have everything integrated, and I think that's a little bit our IP we hold. And we are learning every day. With every client I see, we are learning, because I feel, maybe not as a toddler, but as an intern in my field, like I learn how to manipulate data to have a better phenotypic characterization, which actually fits, which is very important, my clinical understanding of that patient and the clinical understanding of conventional medicine, that it actually makes sense. And I think very often we forget that it has to make sense. And we have to align it with conventional markers, with conventional imaging, with conventional path lab data, that we say, okay, hey, I saw that earlier in conventional medicine, and now I fine-tuned it, I polished it, and I see much, much more and can intervene much, much better.
I love that view of going back into the conventional medicine, because I feel there's such a hype right now in the field where everybody's talking about interventions. But I feel that for interventions to be an intervention, something has to first be wrong to then be fixed, right? And to identify what is actually wrong first, it has a whole process, and that goes from individual to individual. So it's not like, okay, everybody now go and take NAD, because maybe you might need it or I and I may not. And I think measuring all of that for each individual person is really, really important, and sort of like, go back into the conventional ways, find out what actually is lacking to then treat rather than just go for everything and maybe potentially even doing yourself more harm. So I love what are you doing. So, okay, going back a little bit from now into this part of like treating diseases and use the prevention approach. So given that we're not actually trying to heal diseases now in the aging field, and we're actually trying to now, okay, let's see if we can predict something that will actually happen and intervene before it does. How do you actually see this progressing then, since we're not making a statement and a person is not technically sick yet? How do you see this then approaching this next step for prevention?
When I'm talking to clients, the first understanding needs to be there. Aging is not a dichotomous process. Now you're old and now you're young, or you're one or the other, but it's a continuous process.
Yeah.
And the same holds for changes in your blood pressure. It's not that on one day you now have hypertension and therewith you are my patient.
Yeah.
It's a continuous process. The same for the glucose level. Like, it's not that now you are pre-diabetic and now you are really diabetic and now have to throw in insulin and metformin and SGLT2s, etc. No, it's a continuous process. Once patients, clients, but the entire public, and I think also researchers understand, other physicians, like, it is continuous. Let's treat health continuously. Let's treat physiology continuously. I think it's very easy for in clinical practice to have that dialogue with a patient or with a client. However, that does not solve a sort of a little bit now the problem that we don't have yet the ICD codes, which are then actually, which can be used to then also regulate the field. So what we need classification systems. Like, you are in this quartile, or you are in this sextile, or in this, whatever we do, but we need a staging approach where somebody is to then quantify it. And that's the reason why I'm arguing so much for standardization in our field of biomarkers of aging, because if biomarkers of aging are the tools for diagnosing something, I still need cutoff points. I need continuous cutoff points, that if it's a 0.1% or whatever the unit is higher or lower, what does it mean? Yeah. So I need to relate it to something. And that's where we are at the moment. I think the categorization of health, we are going not to a continuous categorization. Of course, you can have endless categories, which is then continuous. But going away from the dichotomization of things. And we need that in our by speaking to clients, we are needing it, speaking to regulators, to speak to health insurers. When should they actually pay or not? It's dichotomous. You are paying or you're not paying. I think that's the decision. And if you are paying, how much do you pay for it? Who is reimbursing it? So these steps need to be made to really implement it into clinical practice.
Yeah. I think, Andrea, you raise a very interesting point here, which is, of course, sort of the dichotomization in context of sort of the payment, but also sort of the regularization and standardization that is required in the field. And I'm almost going on a tangent here. I wanted to get your perspective on, I'm not sure if you heard about the recent most event or that happened at RadFest, where a bunch of people took peptides and felt quite sick in the name of potentially them being longevity peptides, right? And there's almost this sort of market of things which is not very well regulated, and so there's a spectrum of geromedicine, right, where people are doing really good science in clinic, but there are also people that are potentially giving people peptides in very unregulated ways. Are you worried in any ways that this might affect the field, sort of the latter group of people not doing this in a very regulated fashion?
A good point. So first of all, every intervention we are doing and going to do will have side effects. And there will always be a proportion of individuals who is reacting not in the way that we predicted and we don't want to see. So if you are looking at all drug developments, the drugs, even which actually went through the FDA, they can have side effects. This is what we are trained for. Like medications have side effects, full stop. So whatever peptides are being given, also they will have side effects. And it happened on an event, but there is now a different story. I think that the longevity field is like so growing with unregulated interventions, which absolutely needs regulation. And before being regulated, I even think it needs proper science. Like, I have nothing against peptides. Peptides are great. GLP-1 receptor agonists, peptides, like, great, love it. Well studied. Hence, I give peptides in clinical practice. Peptides is just the buzzword for something which might not be good. But peptides are medications I already give decades in clinical practice.
Right.
But what we experiencing in the field of claimed healthy longevity is that we are off-road in terms of bringing evidence to the field and just giving things. If we are off-road, not doing proper studies, then of course the likelihood that there are side effects is much, much higher. And the side effect can be anaphylactic shocks, it can be other reactions, it can be even death. And I'm scared to death that our field is going to implode because of this unregulated actions of the field, that we are going to be so harmed that investors do not want to invest anymore in our field because it's not based on proper science. We have to be science-based, and it can be in an NS1 approach, whatever approach. It doesn't have to be a randomized control trial in the way we did it for 30 years. Whatever we choose, but you have to put it down. We started recently a new journal, and it's called Geromedicine. One of the sections in Geromedicine is called case reports. I think we forgot that normal medicine started with case reports. And I think we are at the stage to just report what we see. And that is also proper science. So everybody hearing this, if you have a case and you want to report on it, please do so. But make sure that before you're doing something, you are measuring something, you're doing the intervention, you are measuring the outcome again. And if it's not the positive effect you wanted to see, it's very valuable for us to read.
Even negative results are critical results.
They're much better. I would like to see many negative results, and I'm very prominent, I think, in the coming years to report negative results because it shows you maybe what not to do, and it guides the field.
Well, absolutely. So people don't repeat the same mistakes, right? Because otherwise it's just we're doing the same things and making the same mistakes, and just wasting money really that we are constantly fighting for research. But just on that point, I have two comments. One thing that I also often see that us researchers do, and is not a criticism in any shape or form, but it's just a lack of evaluation of combined interventions. So, for example, we often look at just exercise or just diet or just enemy. Often people are taking a whole bunch of things all together, and that could have a completely different effect and become again now a problem. And I think we have to step up to see what are the combination of supplements and drugs or treatments that are actually the most effective for people, rather than just focusing one by one. And another thing that I also have sort of like a issue with is that because of the lack of regulation, is we taking now supplements. For example, we hear but for example, and then people are taking, and when you actually go to see the supplement, there is no imminent on it. There is no quality. So it's a huge issue, and it's something that it needs to come with the regulation. So anyway, that was a little bit off notes, but I guess like just to sum up and just to do one final question, we're talking about a lot of people that have access to those medications, and that have access doing those tests, and we know that they're not cheap and they're not easy to track. How do you see us progressing from going from a highly privileged sort of like population to now expanding the longevity field for everybody.
Yeah, very importantly is evolution especially states we have to democratize the health and longevity medicine arena. So the diagnostics and the interventions should just not be for the rich who can pay for it. I absolutely agree with that. In August 2023, we founded the worldwide first longevity clinic in a publicly funded hospital, in Alexandra Hospital in Singapore. And we are treating that clinic as a research clinic. So every patient who is coming is in our observational database that we actually can evaluate what the phenotypic characterizations are of these individuals and if they're reacting to our treatments and how they react. So I think we need, before the real democratization, we need much, much more data to actually see what works, what doesn't work, hence what to implement and how to implement. Very often we say, hey, it should be for everybody. But if that's true, then 100% of the population would have the right to come to a longevity clinic. We are not prepared for that. We are neither prepared for it in terms of content we can deliver, nor we are prepared from a healthcare system. We don't even have the capacity in hospitals. And should it be in hospitals? We haven't even touched on should it be in public health systems and how to deliver it. What is the IT infrastructure to have 100% of the population to be treated?
Yeah.
We don't have the solution. So I don't think that we are there yet for the democratization. Of course, we have to work to it, but we cannot implement it at this moment in time. What we also do here in Singapore is implementation science. So we can open a clinic, but then we have one clinic. And I can tell you, it's real issues. Like, how do you plan people? What do they expect? How do we actually face individuals talking about a biological age? What is their reaction? There's so many, every step is so new that we have to get a step back and first say, okay, what works, what doesn't work? And with implementation science, then to understand what could be the frameworks, the blueprints, which then will change in other countries and other nations, to then replicate what might work better compared to other models. So absolute democratization, but first we need a real footprint, a fingerprint, we need frameworks, we need much, much better science, we need phase 3 trials. Like before giving a SGLT2 inhibitors or GLP-1s, to healthy individuals, we just need big studies. The notion is now that it is going to occur and it's going to occur, but we first need the results before really implementing it. And then we have a much, much better also pole position to talk to health insurers, to talk to the WHO, to get all our ICD codes, to get towards the classifications. And I think it's increasing so rapidly at the moment. This will all occur in the next coming 3 to 5 years.
Yeah. I think, Andrea, you have really sort of laid out some of the barriers quite well in the forthcoming years, right? Whether it's throughout the conversation, whether it's sort of implementing some of these sort of interventions and approaches at large scale, whether it's sort of the regularization and standardization we need, or even the fact that we need to sort of find our right biomarkers. Do you think there's any other sort of big major barrier into implementing precision geromedicine other than the ones that we discussed today?
I think you know me that I'm not a barrier, but I'm an enabler.
We're laying out the barriers so that we can enable eventually.
Yes, that you are summarize all the barriers. I think 5 years ago, I would have never thought to talk to you about having a longevity clinic, not only privately, but also publicly.
Yeah.
Impossible. Like I worked 15 years before, like since 2 decades, to open these kind of clinics. It was never possible. So I think let's bring it towards a positive touch. I think that nobody predicted that our field is growing so immensely. And I think now we are the toddler, we are trying to walk, we are falling again. And there are side effects and there are negative trials, etc. It doesn't matter. But we are there. We are born, full stop. And we are a toddler who wants to walk. And now it's about the direction and about the stability of our field. I would say that there are no other, I think the only barriers are, let's do not screw up, and let's do not make sure that nobody is going to die, and make it bring the field in a condition that we can really grow up.
Yeah, for sure. And of course, an enabler, not a barrier at all. Well, Andrea, thank you so much for joining us today and sharing your insights. It's really exciting to see from your perspective how this field is moving and how it has evolved over the years. And to our listeners, this paper that we talked about today, which we'll tag below in our description, really shows us the future of healthcare, and that the future of healthcare may not just be about sort of fighting the whack-a-mole of diseases of aging one by one, but about precisely managing them. So thank you, Andrea, for coming and enlightening us about all of this.
Thank you so much. One more note, this paper was only possible also because of the visionary of Guido Kurmer. So I just want to acknowledge that we had many discussions and many controversial discussions until we actually, I thought, many words right in that paper.
Yeah, maybe we should have him on as well, Macsue,
Yes.
as our next guest.
Absolutely. Next to reach out. Yeah, well, if you enjoyed this episode, don't forget to follow, share, and leave us a review. And again, check in the notes. We're going to put the paper in there. And we hope to see you next time on Decoding Longevity. Thank you.
Thank you, everyone.
Thank you.
All right. I'm just going to